Welcome to this video on immunofluorescence. In this video, we'll explore what immunofluorescence is, how it works, and its applications in detecting disease. We'll also discuss some of the advantages of immunofluorescence and the current state of the field. Let's get started. Immunofluorescence is a technique that permits visualization of virtually many components in any given tissue or cell type. This broad capability is achieved through combinations of specific antibodies tagged with fluorophores. Consequently, the possible applications in research and patient care are numerous. Immunofluorescence uses antibodies to label proteins or other targets with fluorescent markers in order to identify localization and expression levels in cells and tissues. Imagine you're flying to a city at night. As the plane starts to land, you look out the window and all you can see is the city below you. If there is no light, it'll be too dark to see any features of the land. Now imagine that all of the streetlights in your town used red light bulbs instead of white ones. All of a sudden, we can clearly identify where the roads and highways are because they are marked by a different color than the other lights. We can also use more different colors to mark cars, to identify the traffic flow in the city. As we can track what is happening in the city by different light markers, when we are trying to figure out what is going on in a cell, we can also track what is happening through light markers. But in our cells, there is no lights like cities, so we need fluorescence to mark targets by an immune method. So here comes the technology called immunofluorescence. Immunofluorescence is based on antigen-antibody interaction. Antigen binds with antibody with several non-covalent interactions like hydrogen bonds, ionic bonds, hydrophobic interactions, and etc. There are two variants of these immunofluorescence: direct immunofluorescence and indirect immunofluorescence. For direct immunofluorescence, a usage of single antibody is preferred, which detects the target directly. In indirect immunofluorescence, two antibodies are used, one conjugated primary antibody and a conjugated secondary antibody. Very often, the unconjugated antibody comes from patient samples. Anyway, no matter what kind of immunofluorescence it is, the point of this method is to conjugated fluorescence marker on the target. However, indirect methods is more sensitive than direct ones, because in case of direct method, one antibody only can bind to one fluorescence. It means every target molecule can only have one fluorescence. But in the case of indirect method, two or more antibodies are to the primary antibody which is bound to the target, so there will be more light. The image will be clear, and that's why this method is more sensitive. To make things more clear, let us suppose we have to check the distribution of a specific protein X by direct immunofluorescence. For that, we will add fluorescence with anti-X antibodies on that thin slice of the cell and incubate for some time. During the incubation period, these antibodies will bind to the microtubule present in the cell, and many antibodies can remain unbound. These unbound antibodies can be removed by washing the slide with the help of a buffer. Afterwards, the slide is observed under a fluorescence microscope, which illuminates the slide with a specific wavelength of light. And this light will excite the fluorescence so that it emits colored light at the target. So our target protein can be seen as a colored emission area like this. So in this way, different parts of the cell can be visualized using direct and indirect immunofluorescence. The immunofluorescence method is a method to detect disease and personalized medicine. For example, the Imagine adenovirus kit from Thermo Fisher uses direct immunofluorescence method. It can rapidly detect adenovirus antigen in clinical samples and confirm adenovirus in cell cultures. This direct immunofluorescence assay utilizes a genus-specific monoclonal antibody to detect an epitope of adenovirus hexon proteins, which is expressed in all known human adenovirus serotypes. The advantages of immunofluorescence include clear positioning. This assay can give researchers the clear subcellular localization of molecules. The expression of molecules can be observed directly. high specificity, the preparation of sample can effectively protect the natural structure of the antigen. Excellent sensitivity and amplification of signal employing various microscopy techniques. Many improvements to this method lie in the improvement of fluorescent microscopes and fluorophores. Super-resolution methods generally refer to a microscope's ability to produce resolution below the Abbe limit, a limit placed on light due to its wavelength. This diffraction limit is about 200 to 300 nanometers in the lateral direction and 500 to 700 nanometers in the axial direction. This limit is comparable or larger than some structures in the cell, and consequently this limit prevented scientists from determining details in their structures. Super-resolution in fluorescence more specifically refers to the ability of a microscope to prevent the simultaneous fluorescence of adjacent spectrally identical fluorophores. This process effectively sharpens the point spread function of the microscope. Examples of recently developed super-resolution fluorescent microscope methods include stimulated emission depletion or STED microscopy, saturated structural illumination microscopy or SSIM, fluorescent photoactivation localization microscopy or FPALM, and stochastic optical reconstruction microscopy